All right, so it's really wonderful to see everyone. Pretty low turnout. I think 12:30 on a Saturday is not going to get it done, but it's really not that important, only because we're going to publish this after the fact so people can learn from it when they have time. But it's wonderful to see Ali, Shadea, Rakesh. Thank you for being here. After this, certainly we'll go over any questions you have, but we'll post this online so people have it. So what we're going to be doing every week for eternity, essentially, as long as I have the time to do it, is we're going to have this If You Were My Family Member series that is being sponsored by my companies, but it's not being monetized in any capacity. One of my companies is very much in the patient education space, and someday when we launch, I think people will very much understand what we've been trying to do for patients. And honestly, I think that this will tie in very nicely to that. But when I think about cancer patients, when I think about the people I take care of in the clinic, and there's quite a bit, and I've done it for well over a decade now, the fundamental paradigm for me is that, or the fundamental metric, is that every single patient needs to receive the type of care that I would expect from my mom, dad, brother, sister, child. So I have extremely high standards, and I'm a big believer in patient advocacy. I think a lot of the time, patients need to advocate for themselves, need to understand the nuances of their disease so they can make sure that they're getting optimal care. Now, there's a tremendous number of phenomenal oncologists in the world. This is not by any means intended to substitute for them. It is a supplement. It is a method of patients and family members to be able to really understand certain nuances about their cancer that they're probably not getting from their physicians, only because there's no time to really tell them. So today we're going to just do an overview of breast cancer, stage 4 breast cancer. And really what we're going to do here is it's going to be a little bit like a series. So each week we're going to have an episode related to a particular tumor type and stage. And over time, that mini series related to that stage and tumor type will be complete. So today is episode 1 of stage 4 breast cancer, and it's really going to be an overview of stage 4 breast cancer. These things are intended to be relatively short, and they're intended to kind of build on each other from week to week. So my name is Basem Goueli. For those that don't know me, I've trained at numerous places throughout the world. I did my MD and my PhD at the Mayo Clinic, did my cancer and blood training at Stanford, did my residency at George Washington University in Washington, DC, and spent 4 months at the National Institute of Health. Over my career, I've directed 3 separate cancer institutes, totaling 17 hospitals and 12 cancer centers. We are currently building our own institute, so I've kind of taken myself out of various institutions so I could build my own cancer institute with a vision I've had for over a decade. We have 2 cancer-related companies. I consult for numerous companies myself. I run 2 national clinical, help run 2 national clinical trials for XBiotech, largely in pancreatic cancer, and also have 2 mini-series that 2 editorial series that you can read online. With that said, this is very much intended to be a discourse. So audience participation is absolutely encouraged. Certainly this is in meeting format, not in webinar format. So you can raise your hand. You can literally just talk and ask a question. That's what this series is really intended to do. So let's talk about stage 4 breast cancer. So breast cancer, like any cancer, so cancer is essentially just a cell that grows in an uncontrolled way. So lung cancer is a lung cell, prostate cancer a prostate cell, breast cancer is a breast cell. Now, when we think about breast cancer, essentially the very first question we ask ourselves is, what stage is it, right? And we stage breast cancer 0 through 4. Now there's certainly substages, but essentially let's just talk about 0 through 4 for the moment. So stage 0 to 1 breast cancer is largely restricted to the breast. And in fact, stage 0 really is carcinoma in situ, ductal carcinoma in situ. It's really not invasive. I won't go through that today. That'll be a different talk for a different day. But stage 0 and 1 breast cancer is really restricted to the breast. Stage 2/3 breast cancers are larger. They oftentimes involve lymph nodes, but they typically, well, they have not spread to other areas of the body, right? So breast cancer spreads from the breast to the axillary lymph nodes and then out. So as long as you haven't spread beyond the axillary lymph nodes, you do not have stage 4 disease. Now, if you have spread beyond the axillary lymph nodes and gone to other organs such as the liver, the lungs, the bone, etc., then you really do have stage 4 disease. And the next several episodes of this series will pertain to people with stage 4 breast cancer. Now, when we talk about stage 4 breast cancer, typically what happens in the way you diagnose someone with stage 4 breast cancer is you do imaging, right? You know that the cancer was in the breast. likely know that it was in the lymph nodes. And the question is, where is the breast cancer? Is it truly stage 4? So there's really two ways that we image breast cancer. Number one is we can do a PET CT, and a PET CT is where you give someone labeled sugar. And the underlying principle there is that every cell in your body uses sugar for energy, but because cancer cells divide more often, they need more energy, and in theory should take up more of the sugar. So the tracer, the PET CT tracer, which is radio labeled sugar, should localize to areas of cancer and also can give you a feel for how active those areas are by how much of the sugar they take up. So a lot of us like to do PET CTs. Unfortunately, insurance companies oftentimes reject PET CTs. And that's largely because some of our big national organizations have basically said that you don't have to do a PET CT. I think PET CTs are a lot better than CT scans. Will not go through that today, but I'm a big believer in PET CTs in breast cancer. With that said, it is certainly perfectly reasonable to just do a CT of the chest, abdomen, and pelvis and do a bone scan to really look for breast cancer. Now, some mistakes I do see made occasionally is that when individuals go to look for breast cancer that is spread, they oftentimes, well, not oftentimes, but sometimes will just do a CT scan and not do a bone scan. That's really not adequate. Breast cancer has a predilection for the bones, which we'll talk about in a minute. So you really need to do a dedicated bone scan if you're going to do a CT so you can visualize the bones. Now, if you do a PET CT, which gives you a very good picture of the bones, you do not need a bone scan, right? So you can either do a PET CT or you can do a CT of the chest, abdomen, and pelvis and a bone scan. Those are both perfectly reasonable options. You'll see most people do a CT and a bone scan largely for insurance reasons, again, because of the national organizations I talked about, but I really, really like PET CTs. I'll leave it at that. Now, with stage 4 breast cancer in general, there's no known cure. Now, I have a series online, many of you have seen it, and I'm a firm believer in this, that cancer patients just don't have an expiration date, right? When patients ask me, and they ask me all the time, and they rightfully should, how much time they have, I always tell them, I really don't know. I'm not God. I don't have a crystal ball. I've seen patients do exceedingly well. And really, my thought process isn't in the patient dying. It's in how long can I extend their life with a good quality of life. So that's really the fundamental paradigm, which we'll talk about in a minute, when I see someone with stage 4 breast cancer. Now, with that said, certainly when patients ask me how long they have left to live, I will ballpark that for them only out of courtesy, but I do tell them to take it with a significant grain of salt, because a lot of the data that we use nowadays to really say, okay, you have X amount of time to live is a little bit outdated. It doesn't take into account all the clinical trials you might be on, all the new drugs that are coming out. And so I'm very hesitant when people ask me, unless it's obvious, to tell them how much time they have left to live, because I fundamentally believe there's a lot of hope. Now, I think optimism in cancer and optimism as an oncologist is basically essential. I don't think you can do oncology without being an eternal optimist. You give treatment thinking the treatment will work or hoping the treatment will work. You're not giving someone treatment thinking it's not going to work. So as an oncologist, you probably aren't eternal optimist, but you don't have false hope. And you're not an idiot about it, right? You definitely are pragmatic and you think to yourself, okay, I have a lot of hope here, but I understand what can happen. But the bottom line is I want patients to appreciate that there's a lot of hope, which is really the fundamental premise behind the Cancer Patients Don't Have an Expiration Date series. So when we talk about stage 4 breast cancer, and this is a picture of a PET scan of a patient with stage 4 breast cancer, it's very important to know that bones are commonly involved. Breast cancer has a predilection for bones. That's where you'll see it most often metastasize. So it's something to be aware of. Breast cancer likes to go to bone. 7% of breast cancer will go to the brain, so this is relatively infrequent. It's for that exact reason that unless you have symptoms like headaches that wake you up from sleep or headaches in general, or you have neurologic deficits, in general, your oncologist, your cancer doctor will not get an MRI of the brain, because we're not classically thinking that the breast cancer is going to be in the brain, because it's relatively infrequent that it would go there. Additionally, it's important to note that the lungs and the liver are somewhat commonly involved. Certainly breast cancer does like to go to lungs and liver, but not nearly as much as it goes to the bones. Here you can see a PET CT in a patient with stage 4 breast cancer. You can see that it's in the bones here in the left pelvic bone. You can also see that this is where the breast cancer started. This is in her breast, and you can see a cross section of this here. That's the right breast mass. So this is reversed. It's left here, right here. So the right breast mass where this started. This went to the lymph nodes, which you can't actually visualize here, but it was biopsy proven in the lymph node, and then went to the bones. So here you can see the big mass in the left pelvic bone, which you can see here. And you can also see it went to the left pleura, which is kind of the lining of the lungs, basically the layer of tissue that lines the lungs. And so you can see here, it's in the left pleura, basically the lining of tissue around the left lung. It also went to the right pelvic bone here. You can see it in the backbone. And so you can see that this went to various locations in the bone. Now, when I see a patient with stage 4 breast cancer, my fundamental understanding is that this is generally incurable. So we know that stage 4 breast cancer in general cannot be cured, and theoretically it's incurable. So that's the premise you're starting with as a physician. So the very first thing you're doing is you're thinking, I have to play chess, not checkers, against this cancer. I have to figure out what my treatment approach is going to be. Now, I published this article, it's called Playing Chess Against Cancer, and it's in biopharmatrend.com. You can certainly go read it. But the premise behind that, or the idea, when we see patients with theoretically incurable tumors, not just breast cancer, let's say stage 4 lung cancer, or stage 4 liver cancer, things of that nature, when we have cancers that are theoretically incurable, we typically say to ourselves, okay, we're going to use a first-line treatment. So it's going to be the first treatment we give a patient. We're going to use it until patients progress on it or they can no longer tolerate it. Then we're going to do a second-line treatment. And again, we're going to use that until patients progress on it or they no longer tolerate it, and so on and so forth. So when I meet patients, and this might be unique to me to some extent, because I don't know that a lot of my colleagues do this, I'm typically thinking about the next 5 lines of treatment. So in the very minute I meet someone, I'm not just thinking about today, I'm thinking, how do I get them many, many years of good quality life? So I'm thinking about plan B, already thinking about plan C, plan D, plan E. And in the context of breast cancer, I can tell you a lot of the times, I know my first 10 to 15 plans, including clinical trials. Now, with that said, it's very important to know that it's critical to play chess against cancer. And so to play chess, in my opinion, optimally, you have to really look at this COMET algorithm that I derived several months ago and published. And what that basically entails is making sure that you explore the conventional systemic options that are available to a patient, so conventional therapies. You need to know if surgery is an option, so you think about operations. You need to know about molecular-based options. So we're going to talk about that in a minute, but you have to understand what the genetic mutations are in that cancer. And you've got to know if you can use drugs that target those mutations, or if the cancer has certain molecular characteristics that make it amenable to immune therapy, right? Therapy that's designed to get the immune system to attack your cancer, which we'll talk about in a second. We also think about clinical trials. Now, with stage 4 breast cancer, I tell every single patient, one of your reflexive thoughts needs to be clinical trials. You need to think about it right away because you have a theoretically incurable cancer. So you're thinking to yourself, how do I do better than the status quo? How do I do better than what's available now? And we're going to talk about it in a second, but clinical trials has to be part of your rubric when you think about stage 4 breast cancer, whether it's you or your family member. And then the last thing we talk about, the E part, really is a catch-all. It refers to everything else, but really refers to local regional therapies like radiation, where you basically are radiating a particular location. So you're only addressing that one location, as opposed to systemic therapy that gets everywhere. The local regional therapies only attack the cancer in a particular location where they're acting. So one example of that would be radiation. We'll talk about this a little bit more. But the bottom line is, when you play chess, not checkers, against cancer, what you're trying to do really is make a personalized treatment map. You're trying to say, okay, I have this patient that's unlike no other patient I've ever met with stage 4 breast cancer, because in my opinion, there are no 2 patients that are the same. Even if they're the same molecular profile and they have the same protein expression profile and the same extent of disease, they very likely don't have the same genetic composition. They don't have the same comorbidities, the same other diseases that are present. They just are not the same. So every single patient is different than the other. It's a very heterogeneous population. There's really no homogeneity in cancer. So everyone really needs to have a personalized treatment map. And so to do that, we went through the COMET sort of algorithm. You kind of think about what your options are and you derive a personalized patient map. So if you look here, you can see that this patient has 5 conventional options available to them. Let's say they have 3 clinical trial options. Let's say that when we did their molecular studies, they had a mutation in gene alpha gene beta and gene gamma, suggesting that they can get treatment that is alpha or beta treatment or gamma treatment. And then there's local regional options like surgery, radiation, et cetera, that we think about. And when we kind of look at this collectively, what we say is okay, I've got a stage 4 breast cancer patient that's theoretically incurable. How do I give them an optimal treatment sequence that's going to get them a profound amount of time with good quality? So what you'll do is you'll say, okay, what's the data pertaining to each of these options? How have these options performed in other patients with stage 4 breast cancer? And when you do that, you will then say, okay, I know how these options have performed. Let me now make my optimal treatment sequence for the patient. So you're going to hear me talk about this all the time. If you were at the tumor board we did last week, you saw me do this. If you're at the tumor board we're going to have next week, which is going to be mind-blowing, by the way, you're going to see me do this again and see my colleague Ram do this and Dr. Muskamani do this. And if you come to the lecture next week that we're going to have this part of the next episode 2 of this series, you'll also see me talk about this. I'm a firm believer in this. I think it's very important to really think about what are your contingency plans? How are you going to optimize a patient's treatment sequence? Because I think it has a major impact on how long they live. Okay, with all of that said, let's start with clinical trials. So clinical trials, for those that don't know, and I think most people do, are essentially experiments designed to save lives. Now, I think there's a lot of cynicism regarding clinical trials. I think a lot of people are scared. They're fearful, and understandably so, about getting drugs that may never have been used in people before or are relatively experimental. But please know that the companies, the principal investigators, all the people that have made that clinical trial and made the drug that's involved in that clinical trial and designed it and engineered it and implemented it are on your side. Every one of those people has a fundamentally altruistic motive. They're trying to help you. They're trying to make it so you can live longer with a good quality life. No one is trying to hurt you intentionally. So although clinical trials are experiments, they are coming from a very good place. There's a lot of heart that is entailed in them. Now, there are different phases of clinical trials. So we typically think of them as preclinical, right? So this really isn't the clinical trial part. This is preclinical, where you're looking at the drug in tissue culture, you're looking at it in vivo, you're kind of doing experiments in the lab, and you're also looking at it in animal studies. And then when you kind of feel comfortable with the toxicity in animals, the efficacy in animals, you then take it to phase 1. So phase 1 really is a trial that's designed to see if the drug is toxic or not. Really designed to see if people can tolerate this drug and what dose is optimal to give them for later phases. Now, with that said, certainly part of phase 1 is designed to figure out if the drug works, right? So not only are you using the drug in general to find out if people can handle it and to find out what it does in patients in terms of blood levels, but you're also doing it to see if it works, right? So that's a phase 1 trial. Phase 2 trials are where you essentially take a bigger population of people, you give them the drug, and now you're really looking to see, is it toxic, but mostly looking to see, does it work, right? You're taking a certain number of people, oftentimes say 50 to 100, and you're saying, does it work, okay? If you have a positive signal and it looks like it works, then you say, okay, let's find out if it really works. So you take it to phase 3. Now, phase 3 is where there's a lot more people involved, and now you're typically doing what's called a randomized control trial, where you're giving half the people or some number of people the amount of drug that you want, the drug that of interest, the experimental drug, and you're taking another half of the people, or say 33% of people, and not giving them the experimental drug, and you're really finding out if the drug works or not. And so that's phase 3. And then once the drug is approved, typically after phase 3, we sometimes do phase 4 trials, where we say, okay, the phase 3 study was great, but does it really work, right? So clinical trials, like anything else, are imperfect, right? In the lab, as a PhD, when I did my PhD in biochemistry, we would do experiments in duplicate or triplicate, right? We would do multiple experiments at the same time to ensure that what we were seeing was real, that we didn't just have one experiment that worked and the other experiment that didn't work, right? We would do them in duplicate or triplicate side by side to make sure that what we were seeing was reproducible. You can't do that with a phase 3 trial in general. It's way too expensive. These things cost millions upon millions of dollars to conduct, so you can't really do that. So you might get a positive signal in a phase 3 study with 1,000 people that doesn't really manifest when you look at the general population. Because again, 1,000 people versus 100,000 people, certainly that result may not manifest in the bigger population, just may not extrapolate properly. And then you find that the drug didn't actually work. And that happens actually somewhat frequently, right? So Tecentriq, which is a drug that was approved in breast cancer, was pulled off because they later on found it didn't really work. Panobinostat in multiple myeloma, romidepsin in T-cell lymphoma, umbralisib in CLL, duvelisib in CLL. A lot of these drugs we find after they're approved that they really didn't do what we expect them to do based on the phase 3 trial. So that's essentially a nice summary of clinical trials. But I will say that although there's different phases, the bottom line, the take-home message here is that you have to think about clinical trials if you have stage 4 breast cancer that's theoretically incurable. In fact, if you have any stage 4 cancer that's incurable, you have to think about clinical trials. That has to be part of your treatment, period. If you have earlier cancers, I would still encourage you to participate in clinical trials so we can do better, so you can do better potentially. But the bottom line is if you have a stage 4 incurable cancer, there's no treatment rubric that is satisfactory without involving clinical trials, period. You have to talk to your oncologist about clinical trials. And honestly, what I've seen in my career is that clinical trials can provide hope where there was none. If you look at people who have chronic lymphocytic leukemia as an example, there are people alive today because they were on a clinical trial 8, 9, 10 years ago. And so I think it's chronic myelogenous leukemia, CML. There are people alive today because they were on a clinical trial 22 years ago, right? So these clinical trials have tremendous heart behind them. They are based in altruistic motives, and they can absolutely transform your life. They can provide you hope where there was none. They're an essential component of any optimal treatment map. I ask you to certainly ask your oncologist about these. And if you really want, you can go to clinicaltrials.gov. It's a wonderful website. You can go to the beta version. They have a beta version. Go to the beta version, put in stage 4 breast cancer, and then look at the clinical trials available to you. So I think the bottom line here, the take-home message from this is you have to think about clinical trials. In terms of surgery, so we've done kind of the T part of the COMET algorithm. We're going to do the O part, the operational part. In terms of surgery, in stage 4 breast cancer, there's really no role for surgery. There would be extremely rare exceptions. A rare exception I could conceptualize would be if you had a breast mass and potentially had lymph nodes, and maybe you had one spot in the right lung, and let's say it was a small spot. Your physician in that case might think to themselves, maybe there's a theoretical chance at cure, tiny, but theoretical. Maybe the cancer spread beyond the breast, beyond the lymph nodes, went to that one spot and nowhere else. So they might think to themselves, okay, let me give you treatment, systemic treatment for a little while. Let me do it for 3 months and then do another scan. And let's make sure nothing is going crazy, nothing's growing, things are good. Then I might do another scan 3 months later after I've given you another 3 months of treatment. And now it's 6 months, maybe a year, you say, look, things look really good. Nothing else has come up. Maybe there's a chance at theoretical cure. Maybe we should consider removing these spots. So that's a situation where surgery might come into play in stage 4 breast cancer, but that's extremely rare. In general, please know that there's largely no role for surgery in breast cancer. Of course, I would ask you to talk to your oncologist. Certainly, if you have like one metastasis, say one spot in the liver, well, sorry, I shouldn't say one spot in the lung, maybe you could talk to them about that. But the bottom line is, in general, there's no role for surgery. Now we come to immunotherapy. So a lot of people talk about immunotherapy, and rightfully so. These drugs have absolutely saved lives in many, many cancers. Not as much in breast cancer, which we'll talk about in a minute, but certainly in a lot of cancers, these drugs have saved lives. Melanoma, colon cancer, a lot of patients with MSI cancers, which we'll talk about in a minute, lung cancer. These drugs have absolutely saved lives. The idea behind these drugs is that they are designed to stimulate your immune system to eradicate your cancer. Essentially, wake up your immune system to recognize your cancer cell as foreign and attack it. So you can see here, immune system cell essentially attacking the cancer cell. Now, immune therapy is used in essentially almost every type of cancer. It's hard for me to think of right now offhand, and I do all of cancer, all of blood. It's hard to think of particular solid tumor cancer where immune therapy hasn't at least been used. In addition, when we talk about breast cancer, it does have a role. It's relatively minimal, but there are still studies, numerous studies ongoing, trying to really define if immune therapy is useful in breast cancer or not. We'll come back to that. Now, a lot of you have seen drugs on TV that are immune therapy. You may not know it, but they're immune therapy that we use in cancer. So there's a drug called Keytruda, otherwise known as pembrolizumab. There's a drug called nivolumab, otherwise known as Opdivo. There's a drug called durvalumab, otherwise known as Imfinzi. And there's a drug called Tecentriq, otherwise known as atezolizumab, that you probably have seen a commercial. I've actually seen a commercial for cemiplimab relatively recent, and dostarlimab actually also, and avelumab. I don't know if I've seen a avelumab commercial, but those are the principal immunotherapies that are currently approved by the FDA. There's tons and tons in clinical trial. We won't talk about those today, but the bottom line is you undoubtedly have seen commercials for these drugs that are designed to wake up your immune system to attack your cancer. Now, the drugs I just talked about generally work by blocking the ability of a cancer cell to turn off your immune system cell. Okay, let's back up for a second. When we talk about the immune system, there are numerous cells in your immune system that fight infection, fight cancer, do a lot of the work in that regard, okay? And when we think about it, there's T cell lymphocytes, B cell lymphocytes, there's neutrophils, eosinophils, basophils, there's a lot, okay? Now, when we think about cancer cells, we oftentimes think about T cell lymphocytes that will recognize the cancer cell, attack it, and eradicate it. Okay. Now, one thing that cancer cells do is they can make a protein called PDL1, which you can see here. They can insert it on their membrane, their plasma membrane, their surface. That PDL1 binds to a protein called PD1 on the T cell. When that happens, it essentially shuts off the immune system cell, so it cannot recognize the cancer as foreign and attack it. So we have drugs, the ones I just talked about, that block this interaction. They prevent the PDL1 on the tumor cell from binding to the PD1 on the T cell. So they block this from happening. You can see that here. And by doing that, that essentially allows the T cell to do what it does, attack the cancer cell and remove it and kill it. So the drugs I just talked about, pembrolizumab, nivolumab, atezolizumab, avelumab, cemiplimab, dostarlimab, durvalumab, all of those drugs basically work by preventing this interaction here, so that when they do that, the T cell will kill the cancer cell. With that in mind, what's really important to know is that one thing we do as cancer doctors across the board, doesn't matter if it's stage 4 breast cancer, literally across the board, one thing we do is we will measure the amount of PDL1 that's being made by the cancer cell. Because the higher the amount of PDL1 in general, the more likely you are to respond to immune therapy that's designed to block the interaction of PDL1 with PD1, wake up the T cell, the immune system cell, to attack the cancer and kill it. So we will measure your cancer to see how many of the cancer cells make this PDL1 protein. That's really important to know in general. If you have any stage 4 cancer that's incurable, I want you to go to your physician and say, hey, how many of my cancer cells make PDL1? You want to know what percentage of your cancer cells make this protein PDL1. Good. Now in breast cancer, it's really important because in triple negative breast cancer, we'll talk about, if 10% or more of your cancer cells make PDL1, then we oftentimes will give you a drug called Keytruda, which is designed to block this interaction. And so we give you a drug called Keytruda, which basically prevents PD1 from binding to PDL1. It allows the immune system cell to attack your cancer and kill it. So if your cancer has more than 10% of the cells with PDL1 and it is triple negative, which we'll talk about in a minute, there's a very high likelihood that your doctor is going to give you a drug called Keytruda, otherwise known as pembrolizumab. Now, that's one way we look to see if you will respond to immune therapy. So we look at the PDL1 level. The higher the number of cells that make it, the more likely you are to respond to immune therapy, the more likely your physician is going to talk about it. In stage 4 breast cancer, remember, it's really only the triple negative breast cancer patients with a PDL1 score of 10% or more, so 10% of cells or more making PDL1, that are likely to receive immune therapy based on PDL1. But there are 2 other ways that we look to see if someone might respond to immune therapy. Another way we look is something called microsatellite instability, otherwise known as MSI. So microsatellite instability, a little bit complicated, and I don't want to go through it too much. Just think of it as a measure of how unstable the DNA is in the cancer cell. So cancer cells can sometimes have a lot of DNA instability, which basically manifests as something we call microsatellite instability. Now, the reason that's important is because when the DNA is really unstable, what can happen is the cancer cell can make weird proteins from the unstable DNA. And these weird proteins get secreted by the cell. And the T cell, the immune system cell, says, wait, that's a weird, or the antigen presenting cell says, wait, that's a weird cell. And your immune system, a weird protein. And based on the fact it's a weird protein, we really need to go look at that cell and probably eradicate it. So because of the large levels of DNA instability in the cancer cell, when their microsatellite instability high, you get a lot of these weird proteins. Those weird proteins basically signal the immune system to say, whoa, there's something wrong. Let's go address that cell. Let's go eradicate it. Because of that, patients who are high MSI, so who have a lot of high DNA instability, oftentimes can respond to immune therapy, right? They've got a lot of these weird proteins. So what we do is we use the immune therapy to turn up the immune system, basically turn up the gain on the immune system. So the immune system says, whoa, those proteins are weird. We're going to go attack this cell and eradicate it. Now, this is incredibly important because if you're MSI high, if you have a lot of this microsatellite instability, basically a lot of DNA instability, you are an excellent candidate for immune therapy, like Keytruda, like dostarlimab. Okay, that's really important to know. I will tell you because it can actually save your life. Now, I'll tell you a story. I was directing an institution. I was seeing a patient in a hospital, in one of our hospitals, that came from a very reputable institution that happened to be hospitalized at our hospital. The patient was young, had stage 4 breast cancer, had gone through a lot of different treatments. They had not seen immune therapy despite being MSI high. They did not get immune therapy despite the fact their DNA was incredibly unstable. And sadly enough, that patient died without ever seeing immune therapy when they were known to be MSI high. To me, that's concerning, right? If you're a young patient and you're MSI high and you've exhausted a lot of the typical standard treatments, you really need to get immune therapy. And the reason I mention that isn't because I want to chastise the other institution. I mention that because I want you to be able to advocate for yourself, to look at your molecular profile and say, wait, am I MSI high? Or ask your oncologist. And if you're MSI high, the next question I want to come out of your mouth is, should I get immune therapy? Talk to your oncologist about it. It's critically important. Okay, now, a lot of you probably saw this article in the New York Times about rectal cancer patients. The rectal cancer patients, they gave 12 of them, I think it was 12 of them immunotherapy at stage 2-3 disease. Okay, and so they had 12 of them, 12 patients. I'll answer your question in a second, Rakesh. Sorry, I'm seeing it now. They had 12 patients and they gave them immune therapy at stage 2-3 rectal disease. Normally those patients, we give them chemo plus radiation, we give them chemotherapy, we also do surgery, right? But they were trying to spare them from all of that. So they gave these 12 patients immunotherapy and every one of them had a complete response, right? So when they went back to go look, there was no disease. So they spared them the chemotherapy, the radiation, and the surgery. Now, we will see what happens with those patients long-term, but it looks very promising. It's something I would expect to happen, right? And the reason I expected that result to happen, the reason it wasn't surprising to me, is because those patients had a deficiency in mismatch repair enzymes, okay? So mismatch repair enzymes are involved in repairing DNA when mistakes are made, as they normally are during cell division, which typically happens throughout our lives, right? So the processes in our body are not perfect. Sometimes when the cell needs to divide to basically replicate itself, to replenish cells in our body, the DNA can basically incur a mutation because that process is not perfect. When the cell replicates its DNA to give a copy to each of the daughter cells, sometimes that process isn't perfect. And when there's an infidelity, when there's an imperfection, the body has a number of different proteins that will recognize that, resolve it, and prevent that mutation from being propagated to the daughter cells. And some of those proteins involved are called mismatch repair enzymes. Okay. Now, mismatch repair enzymes are critically important. But the reason I'm talking about it now is because those 12 rectal cancer patients all had deficiencies in mismatch repair. And because of those deficiencies, those people were absolutely set up to respond beautifully to immune therapy. And the reason for that is because you've got lots of mutations in the cancer cell. Those mutations form these weird proteins, like we talked about before. Those weird proteins are recognized by the immune system cell that then attacks the cancer cell and removes it. So the result that was in the New York Times, which was beautiful, I was very happy for those patients, okay? It was a little bit sensationalized only because people who actually work in this field know that that should happen, right? So I've had lots of stage 4 rectal cancer patients where we put them on immune therapy because they had MSI high or they had deficiency in mismatch repair, and they all respond to immune therapy. I've had many patients, I shouldn't say many, roughly 10, because it's relatively rare, roughly 10 patients we put on immune therapy with these defects, and they all responded beautifully. So it was a great result in the New York Times, very happy for those people, but not incredibly surprising. Now, the last thing I'll say here is that when you have a deficiency in these repair enzymes, which we call mismatch repair deficiency, oftentimes that will correlate with a lot of microcellular instability. So these things go hand in hand. Typically, if you're mismatch repair deficient, you will also be MSI high and you'll respond to immune therapy. Listen, I don't want you to take away any of these details. You don't have to. What you need to do is now you need to say, okay, I know I need to ask my doctor about PDL1. And if I'm high PDL1, I need to ask them about immune therapy. I also now need to ask them about MSI status. Am I MSI high? Do I have a lot of DNA stability? Because if I do, my next question is going to be, am I a candidate for immune therapy? It's really, really, really important. Now, the way we check for microcellular instability, and Rakesh just asked this question, is we can do it in 2 ways, Rakesh. So, well, 2 predominant ways. The first way is you can take the biopsy sample. So when we diagnose people with stage 4 breast cancer, we typically biopsy a metastatic site, right? So we have the tissue. We can then we can take that sample and we can do the testing. We send it out to various places. We can test the DNA instability, essentially look for these deficiencies. We can measure the amount of microcellular instability, and we can find out that way. The other way you can do is actually you can take a blood sample. You can also measure the level of microcellular instability that way. So there's 2 ways to do it. Oftentimes in stage 4 breast cancer or stage 4 cancers in general, there's enough of the cancer DNA in the blood that you can just do it from the blood. We like to do it from tissue if it's available. Actually, I should take that back. You can do it either way. They're both good ways to do this. Now, the last thing I want to talk about as we ultimately leave immune therapy for a moment is I want to talk about something called TMB, otherwise known as tumor mutation burden. So tumor mutation burden, just like it sounds, is a measure of the number of mutations in the gene of the cancer cell, right? So just like we talked about before, when you have a lot of microcellular instability, a lot of DNA instability, you typically will respond to immune therapy because you're making these weird proteins. Similarly, if you have a lot of mutations in the cancer cell, you're likely to make a lot of these weird proteins that don't typically exist, right? And these weird proteins then trigger the immune system to say something's wrong, let's go eradicate the cell. So the reason this matters is because if you have a lot of these a lot of mutations in your cancer, a high tumor mutation burden, there is a higher likelihood that you will respond to immune therapy. Again, for the same exact reason we talked about before. You're making these weird proteins, triggers the immune system to say something's wrong and to kill your cancer. And you can see here in this very nice paper published in the New England Journal that the more mutations you had in your cancer, the more likely you were to respond. So a higher response rate. So please, when you talk to your oncologist, if you have stage 4, basically any cancer that's incurable, but stage 4 breast cancer, is what we're talking about today, you need to ask them about PDL1, TMB, MSI. If any of those numbers are high, the next question needs to be, am I a candidate for immune therapy? So you can see that here. I want you asking them about PDL1, MSI, MMR, you can ask if you want, but if they're doing MSI, that's sufficient, and TMB. I need you to ask them that, because the next question out of your mouth, if they say, oh, your tumor mutation burden is high, like it's more than 10 per, basically 10 is the number we look for, or MSI high, right? If you see that, you're going to say, okay, what about immune therapy in this case? What about immune therapy for me or the person I love? That's going to be the very next question out of your mouth, because immune therapy could do amazing things for you or your loved one, right? Or anyone that you're taking care of. And it tends to be very safe. That's the beauty of immune therapy, right? Not a lot of toxicity and highly effective in the right patients. Now, I want to make one very quick comment before we leave immune therapy for the moment. What we do now to try and determine if we think someone will respond to immune therapy, which is basically look at PDL1, MSI, and TMB, is really suboptimal. The status quo right now does not do a great job of this. We miss a lot of people who probably would respond to immune therapy, but we just don't know what to look for. In addition, we have plenty of patients who are PDL1 high and don't respond to immune therapy. So there's a lot more to the story than just what I'm showing you here. And there's a lot of companies, right? A company called OncoHost, OncoSite, Natera, Boston Gene. They're all making different tests to say, well, if you run our test, we can tell you better than just PDL1, MSI, TMB if a patient will respond to immune therapy, which is really, really great, right? Hopefully these tests will be good. Hopefully they'll have a high sensitivity and high specificity, high AUC, and we can use them in the clinic. But right now, our imperfect measures are PDL1, MSI, and TMB. And I don't care if you have stage 4 breast cancer or you have stage 4 colon cancer, stage 4 liver cancer, stage 4 lung cancer, you need to ask about these things here. Okay, so we talked about immune therapy, which is we kind of look to see if you're a candidate for immune therapy by doing molecular profiling. We're going to talk a little bit about molecular profiling now. So what we're doing with molecular profiling, basically looking at the genetic mutations in your cancer, among the other things that I just talked about, is we're trying to see if your cancer has a mutation in a gene that makes a mutated protein that would allow us to attack the cancer using a drug specific for that mutated gene and subsequent mutated protein. So if you look here, for example, you have a patient with a purple gene mutation, so they are a candidate for the purple drug. Patient with the green mutation is a candidate for the green drug. Patient with the orange mutation, a candidate for the orange drug. So we look for these mutations to try and see if there's something about your cancer cell that lets us attack it in a different way. Now, remember, we talked about this earlier. If you think about the optimal treatment map we're trying to generate, right? Think about the options that we have. We kind of go through first line, then second line, then third line, then fourth line, then fifth line. The reason we're doing these tests is because we're trying to figure out if there's an option that's better than everything else. If there's another option we can insert into our treatment map to try and extend the patient's life and make it of phenomenal quality. So this is incredibly important. This is something that is not that new anymore, right? Your physician should be doing this. This should be a reflex for them. If you have stage 4 breast cancer, you need to make sure that they sent something called next generation sequencing, that they sent the entire profile. You need to say, okay, did you send MSI, PDL1, and TMB, and then did you send next generation sequencing? So next generation sequencing is basically where we look at a number of genes in the cancer, okay? And we look at mutations specific for those genes. So you're gonna ask them about that. I'm gonna show you in a minute, but you're gonna ask them about that. Another thing you're gonna ask them about is something called homologous recombination deficiency. So I know this seems like a lot. This is all I'm gonna tell you in terms of molecular profiling. So we can just take away this, it's perfect. But we do look for something called homologous recombination deficiency, which we'll talk about in a second. Now, when we talk about molecular profiling and we specifically talk about next generation sequencing, what we're essentially doing, right, is we're looking at the genes to find out if they have any mutations, okay? This is very important in breast cancer because in breast cancer, patients who are hormone receptor positive, HER2 negative breast cancer, which we'll talk about in a second, 40% of those patients have a mutation in a gene called PIK3CA. And those patients, because of this mutation, are eligible to receive a drug called alpelisib, okay? Alpelisib is a pretty decent drug. It's effective only in patients who have those mutations. It's not effective in patients who do not have PIK3CA mutations. So it's very important, take home message, that your doctor makes sure that they do next generation sequencing. It can be the difference between living and dying. It's not an exaggeration. There are people alive today because they had mutations in their cancer that their oncologist was astute enough to look for and then treat appropriately. So please, please, please make sure if you have a stage 4 theoretically incurable cancer that your physician, no matter the cancer, is looking for next generation sequencing. So in addition to that, make sure they're looking for all the immunotherapy markers, which we talked about. So PDL1, MSI, TMB. And then the last thing I want them to look for is something called homologous recombination deficiency. Now, let me be clear about something. I am pretty confident your oncologists are already doing this. This is not new. This is not specific to Basem Goueli. I'm not, doing something special here. I'm just trying to let you understand what we do so you can advocate for yourself and you can understand what your oncologist is doing, right? And so you can also look at your own reports and say, wait a sec, I have this mutation. Is that useful? I'm MSI high. Can I get immune therapy? It's helpful for you. The last thing I want to talk about in this context is something called homologous recombination deficiency. Now, we talked about how lots of things happen in normal cells that one would not normally expect, right? So when a normal cell divides to form daughter cells to replenish our body, the DNA needs to replicate to give a copy to each daughter cell. Like we talked about, that process is not perfect. And we have a number of different proteins and systems in our body that help repair the mistakes that are made during this process. In addition, what can happen is you can get breaks in the DNA from things like ultraviolet light or cellular stresses, okay? We have a number of DNA repair processes in our body, one of which is something we call homologous recombination. This is a technique or a method the cell employs to try and correct DNA mistakes, okay? Now, I don't want to go through all of this. It gets a little bit complicated. I really don't want to do that. What I will say is that your physician should be testing for something called homologous recombination deficiency. Really, they can test it by just looking for mutations in genes in the cancer cell. But they need to test for this because if you have homologous recombination deficiency, so a defect in the homologous recombination process, you would be a candidate for what we call PARP inhibitors, so poly-ADP ribose polymerase inhibitors. These are decent drugs in the right setting, and the right setting typically is where you have a defect in a gene called BRCA1 or BRCA2 that leads to deficiency in homologous recombination. So what I really want you doing, okay, what I really want you thinking about is I want you to ask your physician if you have a mutation in BRCA1 or BRCA2, because in the context of breast cancer and really throughout all sorts of tumors, you would be a very good candidate for a PARP inhibitor, one of which is olaparib. In fact, olaparib is the one we typically use in breast cancer, but there are other PARP inhibitors, a drug called niraparib, a drug called rucaparib, a drug called talazoparib. But the bottom line is, I want you to ask your physician about all of these different issues. So I don't want you to worry about the details, right? I want you to be your own hero. I want you to be able to advocate for yourself, to just ask your physician about molecular profiling, ask about NGS, ask about PDL1, TMB, MSI, ask about HER2, ask about homologous recombination. Okay? And so if you can ask about that, I think we'll be in very good shape. And so I think that's really important. Make sure you ask your physician about those things. Take this slide with you, right? Or it'll be online. You can just literally write this down. NGS, PDL1, TMB, MSI, HRD. That's it. That's all I want you to take away from this. The last thing we're going to talk about, we're going to end with this, is the protein expression profile of your breast cancer. Your stage 4 breast cancer is obviously very, very crucial. There are 3 proteins we look for, at least now. Someday that might change, right, as we get better at treating breast cancer. But for forever, really, we've just looked for 3 proteins. And actually, breast cancer is something I've studied for a very long time. In 2004, I published a paper on HER2 when I was studying it for 3 years in the lab, right? So I actually studied this protein in the lab and have taken care of stage 4 breast cancer patients for, like 15 years, 14 years. But the bottom line is we look for 3 proteins. We look for a protein called the estrogen receptor. okay? And you can see that here. The estrogen receptor is a protein typically found in the cytosol inside the cell that binds to estrogen and causes the cell to divide in an uncontrolled way. Remember, that's all that cancer is, right? So the estrogen receptor, if your cancer makes a lot of it, will basically bind to estrogen and stimulate your cancer cell to basically proliferate, right? To grow in an uncontrolled way. So estrogen is feeding your cancer if you have a high level of the estrogen receptor. Now, what we do with the estrogen receptor is we basically measure the number of cancer cells that make the estrogen receptor. So if you have more than 1% of your cells, just 1%, making the estrogen receptor, we say that you are estrogen receptor positive. However, there are kind of different degrees of positivity. If only 10% or less of your cancer cells make the estrogen receptor, as long as it's more than 1%, then we say you are low estrogen receptor positive. That matters a little bit because you might be less likely to respond to the treatments we have directed against the estrogen receptor and estrogen in general. We won't talk about that today, we'll talk about it later, but please know that we look for the estrogen receptor to see if your cancer makes it. If more than 1% of the cells make it, we consider you positive. And if you're 1 to 10% positive, we say that you're low estrogen receptor positive. Similarly, we look to see if your cancer cells make a protein called the progesterone receptor. And just like with estrogen, the progesterone receptor binds to progesterone, which then stimulates the cancer cell to divide. So again, if you make the estrogen receptor, your cancer cell is responsive to estrogen. If you make the progesterone receptor, it's responsive to progesterone. And again, just like with the estrogen receptor, if less than 10% of your cancer cells, as long as more than 1%, make the progesterone receptor, then you're considered low progesterone receptor positive. That has slight treatment implications. I'm not going to worry about that today, but the bottom line is just keep this in mind. The last protein we look for, and this is really important, is a protein called HER2. Now, every cell in our body typically makes HER2 on the surface, but what can happen in breast cancer, in about 30% of breast cancers, is that they can make too much of this protein on their surface. That's really important because when you make too much of this protein on the surface, it basically triggers the cell to divide in an uncontrolled way. So we need to know if you make too much HER2 because we have treatments that are designed to shut this protein off. We actually have a lot of them. We'll go through them in the future. I actually just did a whole talk on this last week, but the bottom line is we have a lot of treatments that target this protein. So we have to know if you make the estrogen receptor, progesterone receptor, and HER2. Now, one thing I want to talk about with HER2, I want to kind of hone in on HER2 for a moment. I want to focus on this because things have changed a lot this year with HER2. Things we used to believe, things we used to consider standard of care have absolutely changed. The nomenclature is completely different now. In the past, the way we would determine how much HER2 you had is basically we would do something called immunohistochemistry. This is where we stain your cancer cells with an antibody that recognizes HER2. So we basically give your cancer cells a score, an immunohistochemistry score. We say it's 0, so no cancer cells make it. 1+, a certain number of cancer cells have HER2 on the surface. 2+, more of the cancer cells has HER2 on their surface. 3+, a lot of the cancer cells have HER2 on their surface. Okay, why is that important? Well, it used to be that if you had 0 by IHC, so 0 immunohistochemistry for HER2, you were considered HER2 negative. If it was 1+, you were still HER2 negative. If you were 3+, you were positive. Now, if you were 2+, you were in no man's land. It was considered equivocal. So the way we put you into one of these boxes, HER2 positive or HER2 negative, is we would do something called fluorescent in situ hybridization, FISH. Basically what we would do is we would stain your cells to try and find out how many copies of the HER2 gene it had, okay? Now, the HER2 gene is actually in red here. So you can see that in this situation, there was no significant amplification, not too many copies of the HER2 gene. But in this case, there was a lot of copies of the HER2 gene. So what we basically did in that case is we took a patient who was 2+ by immunohistochemistry. We then did the FISH test. If the FISH test was negative, there wasn't a lot of copies, we said they were HER2 negative. If the FISH test was positive, there were a lot of copies of the HER2 gene, we said, okay, you're HER2 positive. So just to summarize, it used to be that if you were 0 by IHC or 1+ by IHC or 2+ by IHC, but FISH negative, you were HER2 negative. If you were HER2 positive, but positive by FISH, you were HER2 positive. And if you were HER2 3+, you were HER2 positive. Well, that all changed because a drug called Enhertu basically was published. And this drug changed the way we're going to look at this forever. It's never going to be the same. Okay. So people like who were diagnosed with breast cancer 5 years ago now should go back to their doctor and say, wait a second, you told me I was HER2 negative, but was I actually HER2 negative or was I something called low HER2 positive? So let me explain that. What they did recently is they took patients who were estrogen receptor positive with stage 4 breast cancer. Okay. And they were in this range here. They were HER2 1+ by IHC or HER2+ by IHC and FISH negative. So they were either this person here or they were this person here. And they gave them a drug called Enhertu. And they found that that drug did remarkable things for these patients. Basically doubled the amount of time that they had before their cancer got worse relative to standard of care. And that was remarkable. Okay. So what's now happened is they basically said, okay, we don't just have HER2 negative and HER2 positive. We have HER2 negative, HER2 positive, and low HER2 positive. So these patients here, the one that was 1+ by IHC or the one that was 2+ by IHC and FISH negative are no longer considered HER2 negative. They're considered low HER2 positive. Now, many of you will wonder, why does it matter? Well, it matters a lot because 55% of patients who were once told they were HER2 negative are actually low HER2 positive. So I want you to go to your doctor the next time you see them, if you've got breast cancer or the loved one, and say, just so I know, in case something changes, am I actually HER2 negative or am I low HER2 positive, right? Am I HER2 negative, I don't make it at all? Or am I low HER2 positive, I make a little bit and I'm FISH negative? That's going to potentially matter in the future. Hopefully it really never matters for you, but it's something that's good to know. Certainly if you have stage 4 breast cancer, you have to know this. Absolutely have to know this. It's not debatable. You don't pass go. You need to know this. Okay, with all of that said, everyone with stage 4 breast cancer and their loved ones need to know if you need to know your protein expression profile. You just have to know it. You have to know if you're hormone receptor positive. Hormone receptor positive means either estrogen receptor and or progesterone receptor positive. You need to know if you're low HER2 positive or if you're HER2 positive. You've got to know. Go back and ask your oncologist about low HER2. Know that we use a drug for patients who either make the estrogen receptor or progesterone receptor and are low HER2. We use a drug called Enhertu for that. Know that this has a significant impact on prognosis. So if you're triple negative, meaning you don't make the estrogen receptor or progesterone receptor, and you don't make HER2, that has a very different outlook than if you are in any of these other categories. Okay, so what we're actually going to do in the future is we're going to talk about each of these categories separately. So the next 6 episodes of this mini series on stage 4 breast cancer will really tease out what each of these individuals is going to go through. And that's what we're going to do next. And the idea here is we're going to try and create a better chess player. We're going to try and optimize your chances at surviving cancer. So the summary slide here, kind of just to conclude, please know that stage 4 breast cancer is where it's gone beyond the breast and the adjacent lymph nodes. Please know that clinical trials need to be the first thing out of your mouth. You need to say to them, wait a sec, what clinical trials am I eligible for? It has to be part of your treatment rubric. If your physician tells you, we have 0 clinical trials for you long-term, you tell them that you want to go somewhere where you can talk about clinical trials. Now, I want to be a little careful here. There are times when clinical trials are very useful and times when they're not. If you have stage 4 breast cancer that's hormone receptor positive and say low HER2 positive or HER2 negative, the first treatment typically is not going to be a clinical trial. Now, your oncologist is likely very, very smart. They can talk to you about this. We're going to walk you through it in the future. But please know that clinical trials are very, very important. Molecular profiling is very important. Reflexively ask your physician about NGS, PDL1, TMB, MSI, HRD. Go back to this lecture, go back to this discussion, go back to this slide and literally just write them down and say, hey, can you tell me what this was? did you test this? If they didn't test it, make sure they test it. And if they did test it, please, please, please ask them about what the results were. Know your protein expression profile, and please know if you're low HER2 or not. Preview of episode 2, which will be next week. Please know we're going to start building these patient-specific treatment maps in the way I build them. Of course, there's multiple ways to do it. I'm going to try and give you the expert consensus. I'm going to do it based on guidelines. I'm not going to go off script here. I will tell you certain things I think are more art than science. I'll be very clear about that. But we're going to hone in on the patient population that's hormone receptor positive, either estrogen receptor and or progesterone receptor positive and HER2 negative. We're going to walk you through your treatment options. We're going to make you a personalized cancer journey. We'll walk people through it. We'll see you next week. With that, I'm going to stop sharing and I'm going to answer some questions. I think Rakesh had a question here. So let's go to the chat and see where we are. So, okay. So Rakesh is asking, what's the difference between HER2 VE and plus VE? I'm not sure exactly what you're talking about here.
So it's a HER2 positive, HER2 negative, and triple negative. What's the difference?
Yeah, so HER2 positive is where you make the HER2 protein, like we talked about. Okay, so your cancer cell makes HER2 on its surface. Triple negative is where your cancer cell does not make any of the proteins we're interested. So triple meaning 3, right? So it doesn't make the estrogen receptor, it doesn't make the progesterone receptor, and it doesn't make HER2. So we call it triple negative. Now, the treatments are very different between HER2 positive breast cancer and triple negative breast cancer. We're going to talk about that extensively over the next several sessions. But please know that that's the primary difference, and you have to know if you are HER2 positive or triple negative. It's really important. Good, Rakesh. Where does chemotherapy fit into the care option? Excellent. We're going to go through this much later. But in triple negative disease, chemotherapy is right in the front line. It happens right away. Okay, that's why it's important. In hormone receptor positive, HER2 negative or low HER2 positive disease, chemotherapy is typically a little bit later, sometimes in third line, actually, now maybe even fourth line. But chemotherapy is a little bit later. Okay. In patients who are HER2 positive, chemotherapy is right away. You typically get it right away, but you get it with drugs that target HER2. We're going to go through that extensively. I'm going to give you, I think, a beautiful treatment map you can follow. I'm going to go through all the side effects of the drugs you're going to experience. I'm literally going to walk you down a journey for each of these patient populations. And notice hospice was this, no, okay, sorry. That was good question. So the question is, I noticed hospice was the 6-line therapy. That was a mistake. I used this kind of slide for another thing. No, hospice, in fact, I've given patients with breast cancer sometimes 10 to 15 lines of therapy more. The bottom line is, well, I should say more, 10 to 15 lines of therapy. The bottom line is it's always a conversation with the patient. So I always tell people, right? I'm the navigator, but they're the captain. My job is to make sure they understand their options, that they're fully educated about the potential treatment courses we can take. And then they tell me what they want to do. It's their boat. My job is just to tell them the options. And then when they choose their option because they're the captain, my job is to get them wherever they need to want to go safely. So hospice is a conversation that people sometimes want to have, right? And sometimes it's warranted because there's not great treatment options left. The risk just outweighs the benefit. It's a conversation that has to be had. But please know that the 6-line therapy in breast cancer does not have to be hospice. It can be much, much later. I apologize for that. It was a big mistake. I'll take that out. Okay, great. Any other questions? Thank you for this compliment. I appreciate it. Any other questions? Yeah.
One last question, Basem.
Yeah.
I am a big advocate of patient telling their physicians or their oncologists all the questions that you have mentioned. How do we do that? Because when you go and tell them, am I PDL1 positive? Could you do this test? They say, oh, we have a protocol. Then there's a question about like cost. How could you get your point across so they could do all these things for you?
Yeah, Shade, so you're in Canada, so it's a little bit different in Canada, I imagine, just with socialized medicine there. But the bottom line is, I think, number one, you can show them the NCCN guidelines. So the National Comprehensive Cancer Network, which is amazing. If you go to nccn.org, you will see in the guidelines that it certainly suggests they should get these tests. And that's arguably some of the top doctors in the world doing breast cancer, right? They get together and they make these guidelines, which I know you're aware of. You're a pharmacist and phenomenal. The other thing I would say is, look, if this was your sister or your mom or your daughter, you'd insist on these tests. You would absolutely insist on them. It would be a no-brainer. Every single one of them would do it. So I would use that. And then on top of that, certainly you can always enlist a friend to call, like me. Happy to call them and say, can we walk through this? But I certainly think you need to advocate for yourself, and you're amazing at it. You need to tell them that this is what you've been reading, and hopefully they're decent enough to do it. The Canadian health system has its pros and cons. I'm not going to talk about socialized medicine today, but the bottom line is I think that you run into a little bit more difficulty there. And certainly here you run into insurance issues with some of this, undoubtedly, but it's getting much better now.
I appreciate it. Thank you.
Yeah, no problems today. Anyone else? So we will post this online. We will be back next week, hopefully a little bit shorter next week. But I hope people got something out of this. Certainly people who are going to be the tumor board next week, we're going to blow the doors off with next week's tumor board. It's going to be something that MSK, MD Anderson, they have not done this, I promise you. And on top of that, what we're doing next week, you'll now understand it, because you'll see us doing a lot of molecular profiling. So now it'll kind of help you with next week. It's going to be awesome. All right, guys, thank you so much for today. Thank you for joining. We will see you next week. Please have a wondrous week. I wish you all that you wish for yourself.
Thank you, Basem. It was excellent.
Thank you, bud.
Thank you. Great presentation.